healthy or healthier function. Okay, so here you look again at the same cells after incubation with the GcMAF, and you can see that there is a change here. They're becoming more tapering, or we call it ramified. Sometimes we'll talk about that. Cells are becoming more or closer to what we call a healthy neuron morphology. Okay, let's talk about BV2 cells, since we are using those cell lines or tumor cells. BV2 cells are considered microglial cells. Microglial cells generally are macrophages. Macrophages are present everywhere in the body, and they have a very important job, and as the name implies, and the speakers before me said, macro means big, phage means to eat or to engulf. So these cells have one major function, and that's to eat things. Of course, not anything, but they eat the dead cells, for example. They try to engulf or eat bacteria, viruses, tumor cells, and so on. This is their normal, healthy job of macrophages. Macrophages are present everywhere in the body under different names and different morphologies. But we believe or we think that they come from the same family or the same lineage, and that is what we call monocyte macrophage. So mostly in the blood, and then they travel to tissue, and in the tissue they change into different cells. So you find macrophages in the liver, we call them kapha cells, for example. In the kidneys, we call it renal macrophages. In the lungs, and so on and so on. In the central nervous system, we call them microglial cells. And they have different functions there. And also there are differences or different families or types of microglial cells, so it is not just one type. Okay? So think of these as resident macrophages of the central nervous system or the brain, where they have a defense mechanism. And they are extremely important for the immune response or for protecting our central nervous system, i.e. the brain, spinal cord, and so on, from infections. Okay? Now, microglial cells represent a good number of glial population, and that is 10 to 15% overall. They are everywhere in the central nervous system, in the brain, the spinal cord, and so on. And as I said, they have a very, very important job, and that is scavenging. Those macrophages have a different name. Sometimes we call them scavenger cells. Scavenger. This is not a medical term. Scavenger is an English term. And they look for remnant, debris, dead cells, or even if they find bacteria, they try to clear these or engulf these, and you get rid of them. So they are very protective, and you need them all the time. Okay? Microglial cells are also plastic, or there is plasticity, and that is they have structural changes, also phenotypic changes, depending on where they are in the nerve tissue or the central nervous system. As I said at the beginning, there is not one type, single type of microglial cells. That is why there are different types. This is also important. We have to think about the central nervous system. We call it in immunology also. We call it an immune privileged site. Immune privileged site. In other words, it's a special location. There are some special locations in your body where we think that the immune system has to be very, very closely monitored. Like, for example, the ball of the eye. There shouldn't be too much fighting between the immune system and bacteria in your eye, in the ball of the eye itself. Like in the joints, the joint fluids. And also the brain. Because even if there is too much fight, you think of scavenger cells as those are protective, so they are good to be there. But whenever there is an infection, and think of this as a fight between your macrophages and the bacteria or the virus, this fight is not like missiles. You don't shoot rockets on the bacteria and you kill only the bacteria. There's a very good chance also during this fight that will kill also normal, healthy tissue. So, and this is significant. It's important to remember this all the time. Whenever there is an immune response, there is also tissue damage. In TB, tuberculosis, most of the damage that happens inside the lung is not from the bacteria TB, it is from the immune cells. So you don't want the same thing to happen inside the brain. Every time there is an infection, or there is bacteria, or there is something, then you want cells like macrophages to respond in a vigorous way. It has to be controlled and tightly monitored. That is why different glial cells or microglial cells in different parts of the brain look different or perform different functions, so that you can, or their functions are controlled and monitored. Okay. This is important for as I said, for the immunological responses that we may encounter in the brain. Also, when we talk about macrophages in general, macrophages are white blood cells. White blood cells, like neutrophils, basophils, and so on. In origin, they are white blood cells. Whenever you lose white blood cells because of a fight, like if it happens in the blood or in the tissue, your bone marrow will replenish those, will produce more white blood cells. This is not the case also in the brain or the central nervous system. There has to be a certain, there is what we call a blood-brain barrier, and there is control over what can travel through from blood to the brain and so on. So the numbers of microglial cells are controlled, tightly controlled inside the brain. If you lose some of these, it is not easy to get enough cells from the bone marrow at such a short notice. It doesn't work like that. In the blood it works, but in the brain it will take much longer time. That is why those cells are really tightly controlled in terms of number, in terms of phenotypic appearances, and in terms of function. They are very tightly controlled. And they even try to control their number inside the brain itself, away from the blood, so that they can be available if there is a sudden need for macrophages in the site. Okay. When you talk about microglial cells, there are different types of microglial cells. What's important for us in terms of immunity, there are more than these three types, but for immune purposes, we'll talk about those three types: Amoeboid, Ramified, and activated microglial cells. And the three are related to each other, and they can transform from one form to the other form, but they have different functions. Okay. The first type is the amoeboid. Amoeboid, every time you hear the word amoeboid, it means that it doesn't have a fixed shape. It can change its shape. It can move from one place to another. It will follow its cytoplasm. Okay? If you saw, Marco was talking about Arnold and his back. If you saw Terminator 2, the bad guy stood in front of a door and suddenly he turned to fluid. And then he moved from under the door, and then became a man again on the other side. This is amoebic movement. Amoeboid cells can move and change shape, and they can even fit through the very tight places. Amoeboid. And you can think of why. Of course, this is very vital for them, because sometimes the bacteria or virus or tumor, whatever it is in a tight place. So you need amoeboid cells to be able to move into tight places and then go and engulf or eat the viruses. So amoeboid cells have a bulky structure, fat structure. They are mostly present in what we call the perinatal white matter, and there are lots of them, lots of these amoeboid cells in that location, to the point where we call it the fountain of microglia. There are lots of cells there. They move freely in that region, and of course this is very vital for them. They move because they are amoebic cells. They move using their amoeboid or false feet, and this way they can wander around inside the central nervous system, okay, and work as scavenger cells, removing debris or dead cells or dead bacteria or whatever looks abnormal. So this is their job. They are phagocytic cells. They remove debris. But keep in mind that those cells do not do what we call in immunology antigen presentation. Of course, if you want to talk about something called antigen presentation, you need lectures about antigen presentation. But what macrophages do, they do two jobs. They do two things. They eat antigens. Think of this as bad bacteria or tumor cells or dead cells. And also they present part of these to the rest of the immune system. In other words, they educate the immune system about the nature or the structure of the antigen that is invading the cells. We call this antigen presentation. This is very important role played by macrophages. Now, most macrophages can do that in other tissues. They present, they eat engulf the antigen, and they also present parts of it to other immune cells for education and for activation. Those amoeboid cells do not perform this function. So you ask yourself, I mean, we have a specialization here. I mean, the same cell in other places will do all these functions. A macrophage in the blood or in the tissue will eat cells and also will do antigen presentation. Here we have a specialization. Some cells will just remove the debris, the dead cells, for example. And then they should, after that, do antigen presentation, but they don't do that here. There is specialization. Why not? Why not do that? It will be more economic. It will be more effective, considering place and space and all these things. But keep in mind that if antigen presentation happens, there's a very good chance that there will be a strong immune response happening after that. So we have to understand the function here. If you have a bacteria that's alive, what do you want to do to this bacteria? You need to kill this bacteria. But if you have dead cells, what do you want to do with the dead cells? Just remove it. You don't need to kill the dead cell. It is dead. So debris or old tissue, damaged tissue, or old tissue and so on, it needs just to be removed. You don't need to kill it. So this is really something that you will not see in macrophages except in the brain. In other places, macrophages will remove and also they fight, even if it is dead. They will start producing an immune response. Here we don't say that, we see that. And this will minimize the damage to the brain that can happen. Okay, so this is extremely important as a function, because as I said, you don't have the luxury to make fights every time there is a problem or there is debris in the brain. Okay, the other function of amoeboid cells is what we call rewiring of the brain. These branches and connections between the neurons. This is the way of communication in the nervous system. This is the way of communication between the nervous system and the rest of the body. More connections, more dendrites, more axons, branches of these neurons means you have a healthy brain. And this is one of the functions of the amoeboid cells. Okay? The second type of microglial cells is what we call the ramified. Ramified means tapering. Tapering. They are thin cells and they have branches. And these are present everywhere in the brain, in the entire brain. And they are usually present where you don't have any foreign material. Yeah, you can think of these as resting cells. They are not for the fight. They are not for removing or producing an immune response. Okay? But what's peculiar about them is that they have very active branches. And these branches, they keep moving around or touching the surrounding of the cells, looking for or trying to sense, or we call it surveillance function, look for whether there is anything abnormal, any bacteria or any abnormal tissue or anything happening. So these are important for sensing. They are motionless. They are not actively moving like the amoeboid cells, for example. So you don't see them actively moving. We call them motionless. And even if they move, it will be at a very, very slow rate. Okay? These cells cannot phagocytize or phagocytose foreign material. Okay? Well, I said that these are microglial cells. Yes, microglial cells, but they are specialized. So amoeboid can remove, but they don't present. These cells cannot phagocytose at all. So what's their function? It's just their surveillance. They're sensing the environment. Okay? And whenever the environment is safe and there is nothing unusual happening, there is no infection, those cells are active and doing whatever they are doing. And then whenever there is an infection, other cells, other microglial cells will do the fight. The third type is the activated, and as the name implies, these cells, once there is an infection or when there is a foreign antigen, for example, they can be activated. And those are the typical immune active macrophages of the brain, or the microglial cells. Okay? They are large in shape, They are the ones that perform what we call a typical phagocytic antigen presentation functions. They can even produce inflammatory substances or mediating substances. They are capable of phagocytosing or phagocytizing antigens, and they are also capable of presenting these antigens to the rest of the immune system. So they are phagocytic and they are antigen presenting, okay, different than the other ones. So these are fully functional, typical macrophages that we know, and they can activate the rest of the immune system, the immune cells like the T lymphocytes or so on, okay? And they are activated usually whenever there is an infection, for example, or there is a tissue damage in the brain or in the site itself. Keep in mind again and again, these produce what we call pro-inflammatory mediators or signals. This is important because this will attract the rest of the immune system. This will activate the immune system. At the same time, this is a bad sign because if this happens for a longer period of time, there will be more and more damage in the brain. Something we see sometimes in neurodegenerative disorders, for example. So, what is the role of these microglial cells in neuroinflammation? Okay, so every time you activate you use the activated microglial cells or activate the microglial cells, there is a change or shift in gene expression, and this will, of course, translate into production of what we call inflammation, and manifested in the production of neurotoxic mediators. The cells preparing to fight, to kill. So it will produce what we call neurotoxic mediators. Of course, these are good if there is an infection because they will help eradicate bacteria, viruses, tumors, and so on. But again, if there is a chronic inflammation, for example, chronic neuroinflammation, then this can also affect the tissue of the brain, and then lots of mediators here in the brain itself results in more and more damage to the brain itself. And this can contribute in the later on, downstream, if it continues like this, can contribute to the more and more damage and what we call neuronal death in the tissue. Okay. Now, there is a very tight balance when you activate these neurons or when you use them, Amoeboid, Ramified, and activated. If you keep the balance shifted towards, most of the time, there is no need for an immune response. If we always keep pushing towards what we call the resting ramified neurons, then this will contribute to what we call the homeostasis of the brain, the balance, the normal, the health of the brain itself. And there is no neuroinflammation affecting the brain cells. Okay. What happens if you activate the cells, pushing them all the time towards the active or activated form, then this can contribute to more and more damage. And this has been seen or documented in like Alzheimer patients. Okay? And actually this is a model. Long activation of microglial cells or pushing them towards the activated form can contribute to what we see, the symptoms we see in Alzheimer's disease. And this has been published and documented. Actually, one way to control these neuroinflammation is to shift the balance from more of the activated form, if you can, into what we call the ramified form. And if you achieve this in patients who have chronic neuroinflammation, then probably you can restore a healthy neuron phenotype and healthy function. Now, in culture, if you put BV2 cells, these are mouse microglial cells. If you put them in culture media, the cells are always activated. These cells are shifted towards the activated form. Why? Because the medium itself contains the nutrition or the medium we put these cells in contains factors that can push towards the activation of the cells. I mean, this is normally present in normal culture media, like if you use the RPMI 1640, for example, with fetal bovine serum and so on. they contain some of these mediators that will help in the activation or help or push towards activating the microglial cells into the activated form. But if you don't, if you leave these cells, or if you leave them as control, if you don't add anything, they will be activated. Now, let's see what happens if you add GcMAF. Now, in this figure here, you see two pictures. The one on the left is the typical ramified form. This one. You can see a thin body, lots of branches for sensing, the typical ramified form. Now, the one on the right is one that is taken after a traumatic brain injury, which is the activated form. And you can see that there is a phenotypic and appearance difference between these two. You don't even see lots of these branches coming out of the cell. So here, these cells are actively involved in phagocytosing, in eating, engulfing the damaged material, and performing their normal function, and that is phagocytosis. Now, let's see what happens when you use GcMAF with these PV2 cells. Now, these are the cells in culture without GcMAF. You can see their appearance here. And then you treat the cells with GcMAF, microglial cells, GcMAF, and then changing the concentration, increasing the concentration, and then we see what happens. Now, this is GcMAF at a concentration of 8 picomolars, 24 hours. And remember the original one without, and you can see that cells are changing their phenotype, they're changing their shape. You start seeing the branches, these branches. It is more going towards the ramified form. GcMAF 8 picomolar, 24 hours, this is the larger magnification. Here, looking at 800 picomolars at 24 hours. Okay, 24 hours at larger magnification. You can see that the cells are more and more developing branches, and they are even connecting to each other, okay, which is a healthy sign. Closely looking at two cells at a higher magnification, and you can see that there is connection established between them. Remember, in the one just in culture, we did not see that many connections or neurites, and we did not see this link between the cells. Okay. Now, also, if you use GcMAF with some of these microglial cells, you can, or they can change into the amoeboid cell type. And amoeboid cell type, remember that they can phagocytose, but they do not present antigens. Okay? And again, amoeboid cell types are important for the rewiring, establishing connections. When cells change into the amoeboid, especially after brain damage, they will help to re-establish connections between the different parts of the brain. So it's a repair mechanism also. So some of these cells are important for protection, and at the same time, other cells from the same family, they are important for the rewiring of the brain after or at the end of the immune response. So they help in what we call the rebuilding of the brain. Okay? So here you see the bulky cells like these, and these are what we call the amoeboid cells. Okay, closer look here. The ramified usually are thin, and they have lots of branches. These are mostly amoeboid cells. Okay, so GcMAF in a way helps to rebuild the brain, helps to rebuild the connections between the neurons of the brain, and this will contribute to the health of the central nervous system. Okay. The final slide here of this first part is, let's look at the two types of cells together. Here we have a co-culture of glial cells, neurons and microglial cells. You can see here we have the neurons and the glial cells. Okay. This is controlled, this is without GcMAF. And now after you add GcMAF at low concentration of 8 picomolars, you can see the difference here. There is crosstalk between glial cells and the neurons, and it's the picture speaks for itself. Big difference. And this connection, as we said, in the central nervous system, this is what matters. More connections, more crosstalk between the neurons. Okay. The second part of my talk will be looking at megalase.
Can I suggest that we break for coffee at this stage, and then we'll have the second part of your talk and your questions.
Okay.
So we'll take a 20-minute coffee break, and then we'll resume.
Made it, Andrew.